In this video, naive T cells, labeled red, interact with dendritic cells, labeled green, in the T cell zone of a lymph node. The timestamp displays the minutes before and after the injection of a peptide antigen recognized by the T cell receptor of the T cells. When we repeat the movie at a slower speed, note that the T cells initially migrate freely between dendritic cells within the T cell zone. However, within the first 10 minutes following the injection of antigen, the T cells become activated and establish stable contacts with individual dendritic cells, as indicated by the white circles. These stable contacts are mediated in large part by LFA1 ICAM interactions. In a close-up view, we see the initial interactions of the T cells with dendritic cells. After the T cells recognize antigen, they start to engage the dendritic cells. Note how the T cells continuously move across the surfaces of the dendritic cells, maintaining close contact with both the cell body and the long arm-like processes called dendrites. Once antigen-mediated contacts are established, the interactions between individual T cells and the same dendritic cells may persist for hours or even days. Here we see the prolonged association of T cells with dendritic cells that has continued for 6 to 7 hours after the injection of peptide antigen. During this time, in response to signaling between the cells, the T cell will begin to develop its effector function. In this experiment, as before, dendritic cells are labeled green, and T cells that recognize the injected peptide antigen are labeled red. However, there is also a separate clone of T cells that do not recognize the peptide antigen, and they are labeled yellow. After the injection of the peptide, note that the red T cells that can recognize the antigen rapidly establish stable interactions with dendritic cells, but that the yellow T cells that cannot recognize the antigen continue to move randomly and fail to establish stable interactions with the dendritic cells. Having failed to recognize their cognate antigen, the yellow T cells will exit the lymph node within minutes to hours. The red T cells will remain associated with the dendritic cells for hours or days and exit the lymph node as effector T cells after undergoing several cycles of cell division and differentiation.